This talk is going to be a sort of personal history of research in cell death. So cell death bench to bedside. To get the first slide going now. So all of us have 46 chromosomes, and it was only discovered in the 1960s that that's how many chromosomes humans have. Up until then, it was thought that humans had 48 chromosomes, and once they worked out techniques for banding them, they were embarrassed to find out that we each have 46 chromosomes. We get 23 from our mothers and 23 from our fathers. And now each chromosome, and all of our cells have all 46 chromosomes except the red cells, each chromosome is a single double-stranded twisted molecule of DNA. So it's just 46 molecules of DNA that we have within each of our cells. Now, if you stretched out those chromosomes, those 46 chromosomes, and laid them all together end to end, they would stretch for about a meter, or actually closer to 2 meters long. Our entire genome, so altogether those 46 chromosomes have 6 billion letters, and these are the letters known as GATC that encode the human genome. So every time a cell divides in two to make a daughter cell, those 6 billion letters have to be copied. And because over 1 million cells in our bodies divide to split in two and make a copy of their DNA every second, it means that every second since the day we were born, our bodies make over 2,000 kilometers of DNA. And although the chromosomes are very long, so they're single thread, single molecules, they're also very thin because they're only a few atoms wide, and sometimes they can break. Now, when chromosomes break, and in fact, in order to copy them, you've got to unwind them, and that involves breaking the DNA and rejoining them together again using enzymes called helicases or topoisomerases. But when chromosomes break, the cell tries to repair them by sewing the ends together. But if 2 chromosomes break at the same time, sometimes a cell will make a mistake and join one chromosome to another chromosome. It gets it wrong, and that's called a chromosome translocation. Now, in 1978, this woman, Janet Rowley, was looking at the chromosomes of patients who had a cancer of the white blood cells. These are the B cells, the antibody-producing white blood cells. And this particular malignancy of the white blood cells is called follicular lymphoma. So malignancies of the white blood cells could be leukemias or could be lymphomas. So she was looking at the chromosomes, and in almost every case of follicular lymphoma, she found the same abnormalities involving chromosomes 14 and chromosomes 18. So if you look at these chromosomes here, you halt the cells in the middle of cell division when their chromosomes are compact, and you add chemicals that give them the bands, and that allows you to recognize the 2 chromosome 1s, the 2 with a pair of second chromosomes, and so on. And you can see here, these must be the chromosomes from a female because there's 2 X chromosomes and there's no Y chromosome. But Janet Rowley noticed that in cases of follicular lymphoma, they always had this same abnormality between chromosome 18 and chromosome 14, where a little bit, the tip of chromosome 18 is stuck on the end of chromosome 14. So this chromosome 14 is abnormally long, and this chromosome 18 is abnormally short. Now, this close association between this very particular chromosome translocation of follicular lymphoma suggested that maybe it's not just coincidence. Maybe there's something causal, and this chromosomal abnormality can give a clue to the original cause of follicular lymphoma, because the normal cells in the patient don't have that particular translocation. So Yoshida Sugimoto, who was working in Carlo Croce's lab in the United States, cloned the gene next to the breakpoint on chromosome 18 that's carried over to chromosome 14, and he called the gene at the breakpoint BCL2 for B-cell leukemia gene number 2, because the cells with the translocation and the cells in follicular lymphoma are B-cell malignancy. They're the antibody-producing cells, and he was looking at cloning genes for the first chromosome translocation he found in B cell, he called the gene at the breakpoint BCL1 and BCL2 and BCL3 and so on and so forth. But this was the second one he identified, and he called the gene at the breakpoint BCL2. So the question is BCL2 a cancer gene? Does that translocation that puts the BCL2 gene onto chromosome 14 where the antibody genes are, does that cause the cell to make lots of BCL2 and cause that normal cell to turn into a malignant cell? So is BCL2 a cancer gene? And there was precedence. There's another leukemia called chronic myeloid leukemia. And in that disease, there's a typical chromosome translocation where there's a tiny chromosome form called the Philadelphia chromosome. And at the breakpoint in the Philadelphia chromosome, there was a gene called ABL, and ABL was known to be an oncogene, a cancer gene that drives cell growth. So the first thought was maybe BCL2 is also a cancer gene, maybe it also drives growth of a cell. But the question is, well, how do you find out if BCL2 is really a cancer gene? And the way to test it, or one powerful way to test it, is to make a transgenic mouse. Now, the first transgenic mouse that was made was a transgenic mouse that expressed a gene, another cancer gene called the MYC gene. And interestingly, although the MYC gene was first found in a leukemia virus in chickens, the MYC gene is also another gene that's translocated in a different kind of lymphoma called Burkitt lymphoma. And MYC turns out to regulate cell growth. But to prove that MYC was an oncogene, these researchers in Phil Leader's lab turned on the MYC gene using a promoter that it turns on the MYC gene within breast tissue. And these mice these transgenic mice carrying an extra copy of MYC that's switched on in their breast tissue develop breast cancers. So this by making transgenic mice, it's a very powerful way to show that a potential oncogene really is capable of driving cancer development. So that mouse, one of the first cancer prone mice developed, was called the oncomouse. And because they were at Harvard, they patented these mice, and they sought patent protection in the United States and several other countries, but interestingly not in Australia. And that will come up later in the talk. So when I joined WEHI to do a PhD, I was in Jerry Adams' lab, and Jerry Adams and his wife Suzanne Corey had also developed transgenic mice. This was they produced these ones later than the oncomice I showed you before, and this paper was published in Nature at the end of 1985. And they took the MYC gene, the same gene as Phil Leader's group did, and they turned it on under control of a promoter that works in B cells, so in the B lymphocytes. Now, these mice were made in Washington, the United States, by Roger Parmenter and Brinster, but the constructs were made here in Melbourne, and but these mice all developed a lymphoma, once again proving that the MYC gene really is an oncogene. And then they sent the mice out to Melbourne for further analysis. Now, Jerry Adams was my PhD supervisor, and he thought, well, let's make transgenic mice expressing BCL2, just like the MYC mice, to see if they would also develop lymphoma. And he wrote to Michael Cleary. This is a picture of Michael Cleary, who was at Stanford, because Michael Cleary, like Yoshida Tsujimoto, had cloned the BCL2 gene. So Jerry, my boss, wrote to him and said, can you please send us this BCL2 gene, the gene translocated in this 1418 translocation of follicular lymphoma. And Mike Cleary sent him the BCL2 gene in a little test tube, but he also wrote this letter. He says, dear Dr. Adams, I'm enclosing a subclone, and it was in this single-stranded viral sequencing vector, M13, and but he said it encodes the entire reading frame for the BCL2 polypeptide. he said, I gather that you're interested in determining whether BCL2 may play a role in B lymphoid tumorigenesis in the ENU-MYC transgenic mice. So to see whether BCL2 and MYC might cooperate. But he said, I will be very interested to learn of your results regarding this issue. But he said, part of my current research involves introduction of human BCL2 gene into the mouse, in other words, making transgenic mice, under the control of the Ig heavy chain enhancer. But therefore, my only request in sending you this clone is that you refrain from carrying out similar experiments. So Jerry had the BCL2 cDNA, but he couldn't carry out the experiment to make the transgenic mice because that's what Cleary said he wanted to do, and it would have been a bit rude to do the same experiment that Cleary said would be in direct competition. So, Plan B, and it turned out Jerry had a new PhD student in his lab, so he gave that new PhD student, in other words, me, the tube with BCL2 in it, and said, well, can you figure out if it's a cancer gene? But of course, you can't make transgenic mice. Now, so I thought, look, I'll take a different route and I'll use cells from this cell line. So this is a line of cells called FDCP1 cells, and these cells were developed by Don Metcalf at the Walter and Eliza Hall Institute because these cells grow in the presence of interleukin-3, a cytokine, a growth factor for white blood cells. And if you take away interleukin-3, then the cells all drop dead. So this green stain and this red stain indicate cells that have died. Now, Don Metcalf used this cell line as an assay so he could purify and assay sources of interleukin-3. So it was very much a tool cell line that indeed allowed him to purify to absolute purity interleukin-3. So here's Don Metcalf, who developed that cell line. And the next picture is Wendy Cook. Wendy Cook had taken these cells and she'd infected them with a mouse virus that expressed this a viral form of this oncogene called v-ABL. Now, I mentioned ABL before because it's a variant of ABL that's involved in chronic myeloid leukemia, a similar sort of white blood cells to these white blood cells. Wendy Cook had shown that if, normally if you take away interleukin-3 from these cells, then they drop dead. But she showed that if you put v-ABL in these cells, and took away IL-3, they'd keep growing. So ABL is an oncogene that stimulates cell growth and can replace the need for any growth factors. So I thought, well, this seems like it would be an ideal test to see if BCL2 could also stimulate cell growth and take away the need for growth factors. So I thought maybe I'll put BCL2 in that same cell line. And to do that, I got the help from Ishwar Hariharan. Ishwar was the PhD student on the bench next to me, and he cloned the BCR-ABL gene from chronic myeloid leukemia, and he'd put it into a vector he developed called the Zen retroviral vector, and he'd shown that he could reproduce Wendy Cook's observation using the BCR-ABL fusion gene. So, so he encouraged me to put BCL2 into his Zen vector, and here's a picture of the Zen vector with BCL2 in it. You can see that the Zen vector has an antibiotic-resistant marker, so you can easily select cells that have taken up the retrovirus, and this virus will express the BCL2 gene. So I put BCL2 into some of these FDCP1 cells, and I took away growth factor, and nothing happened. I did the control where I put in Ishwar's ABL virus, and the cells were able to grow in the absence of growth factor. But when I put in BCL2, I never got any colonies unless I added growth factor. But as I'd look at the cells down the microscope again and again and again in soft agar to see maybe there's a colony growing, and I never saw a colony, but sometimes I'd see a pair of cells. And when I looked at the control cells, they were all dead. But when I looked at the ones with BCL2, I'd often see bright, shiny cells. So they weren't growing, but it appeared that they hadn't died the way the control cells did. So then I took 20 clones of cells that I selected for resistance to neomycin using the neo-selectable marker. And I took those 20 clones of control cells, 20 clones of cells with the BCL2 virus in, and I grew them up, and then I took away growth factor, and these are the results here. So these are, it's actually 18 individual clones of cells, and these ones have the empty virus, and these ones have the virus that has the BCL2 insert. And if I added interleukin-3, then in these wells, I just see thousands and thousands and thousands of cells. But in the controls, I counted how many V for viable cells there were and how many dead-looking cells there were. And you can see in all of these independent 18 clones, there were no live-looking cells, and all of the cells were present, but they all looked like dead corpses. When I looked at the cells that had BCL2, I could often see some dead-looking cells, but in almost every case, there were some cells that looked quite viable. They looked bright and shiny. And importantly, if I added back interleukin-3 to these cells, then the cells were able to proliferate once again. So that meant BCL2 was different from the other cancer genes. It doesn't stimulate cell growth, but it prevents cells from killing themselves. Because after all, interleukin-3 or these cytokines don't provide nutrients to the cells. The cells, whether they've got cytokine or not, they've got amino acids, they've got glucose, they've got oxygen, they've got everything they need to survive. Interleukin-3, the cytokine, provides information. And the cells that don't get that signal from interleukin-3 must be killing themselves, activating an inbuilt self-destruct program. But BCL2 was somehow able to block that cell suicide process. This is a picture of some control cells when you take away growth factor, and you can see cells eventually lose membrane integrity. They also get condensation and compression of the chromatin within the nucleus in many of the cells. But the cells expressing BCL2, the cell membrane's intact, the nuclear membrane's intact. You still see heterochromatin, but most importantly, if you add back interleukin-3 to these cells, they're able to proliferate once again. So BCL2 did not allow IL-3 dependent cells to grow or proliferate, but it prevented them from dying when this growth factor was removed. So it used to be thought that growth factor binds to a receptor and it gives a signal that it stimulates cell proliferation and survival. And if you took away growth factor, there was no signal and no proliferation and no survival. But these experiments meant that there must be a branch downstream somewhere of the cell receptor, and that branch goes to stop a default death pathway, and it also stimulates proliferation. And if you put in BCL2, BCL2 can block that default death pathway, but it has no effect on proliferation. If you don't have BCL2 and you take away growth factor, then this default death pathway activates and the cell kills itself. So once again, these experiments meant that there are distinct pathways that control cell division and cell survival. BCL2 specifically acts in the cell suicide pathway, but doesn't have any effect on proliferation. And it also means, because of this association between translocations that activate BCL2 and cancer in humans, and follicular lymphoma is a very common cancer, also chronic lymphocytic leukemia also has high levels of BCL2, it suggests that if you inhibit cells' ability to kill themselves, that can also eventually lead to the development of cancer. So I still wanted to do the transgenic experiment to make BCL2 transgenic mice. So I talked to Jerry and said, look, you know, can we talk to Cleary and ask him to give us permission to do that experiment? And Jerry said yes. And so we went into his office and we rang up Mike Cleary at Stanford in the United States and said, look, we'd still like to make BCL2 transgenic mice. Will you give it give a nod of assent? And Mike Cleary said, well, he'd made BCL2 transgenic mice and he'd seen no phenotype. So he said, yes, we were free to go ahead and do that experiment. So then I started making transgenic mice. And this shows, you know, the first lot of founder transgenic mice. This is making the DNA that's then put into a glass pipette that's injected into the mouse embryos. And the person who did the injections was Mary Bath. Now, although her name is Mary L. Barth, everybody calls her Sue, except for me, and I call her Tubbs because her surname's Barth. And she was a whiz at making transgenic mice. She learned that technique in the vet school with Mal Brandon, but she'd also been a technician working for Jacques Miller and for a lot of luminaries in immunology. But she really got the transgenic effort underway at the Walter and Eliza Hall Institute. You remember those MIC transgenic mice, for example, they were actually made in Seattle. Now. And so here's an annual report from WEHI where it says that how, so this is the 1988-1989 annual report. We've constructed emu BCL2 transgenic mice. We've made over 25 primary transgenic strains. And this was quite difficult because there were no monoclonal antibodies against BCL2. So it's very hard to tell if BCL2 is actually being made from the transgene. The only way to do that was to do northern blots on the mice. And northern blots, for those of you who've ever done them, they're really, really tricky. These are before the days of PCR and so on. But we made over 25 independent strains of mice. And this is from my PhD thesis, where it turned out that those mice didn't develop lymphoma. You could take out the lymphocytes from those mice and they survived well, even in low serum conditions, but the mice didn't develop lymphoma. If we crossed them with the MIC transgenic mice, then they developed leukemias extraordinarily rapidly. So clearly it was functioning, but although the mice didn't develop early lymphomas, they did die early of a kidney disease. And this is a picture of a section through the kidney, and you can see immunoglobulin deposits in the renal tubules. Now, my colleague Andreas Strasser then joined the lab just as I left to go over to Stanford, and he analysed the mice from an immunologist's point of view, and he discovered the mice were suffering from a disease that resembles SLE, or systemic lupus erythematosus. And that's the hypothesis is that's because normally, so that we don't develop an autoimmune disease, we normally cause our lymphocytes to undergo programmed cell death or to kill themselves, and that gets rid of the autoreactive cells. And if the mice didn't get rid of those autoreactive cells, then they develop this autoimmune disease, SLE or lupus. So cell death is not only important to prevent the development of some cancers, it's also important to prevent the development of autoimmune diseases. So failure of cells to die because they have too much BCL2 seems to promote both cancer and autoimmune disease. But I'll just back up a little and talk in more general terms. Right? So there are at least 3 properties shared by all kinds of living things. So what's your definition of life? One thing is that all living things are made of cells. At least some members of all living things, some of their cells can reproduce. If none of them can reproduce, well, they're not going to live for very, very long. And the other thing about living things is all living things can be killed. All of them are mortal, i.e. they can die. Now, it's self-evident that cells will die if some process needed to keep them alive is inactivated. So if you deprive them of nutrients, or if you put them in sulfuric acid or strong toxins, or put them in a Bunsen burner, or something internal happens, like proteins don't fold correctly, or you accumulate macromolecules, all cells can be killed. So all cells are mortal. But what is perhaps surprising is that of the 1 million cells produced in our bodies every second, since the day we were born, almost all of them will die, not because something kills them, but because they kill themselves. In fact, in our cells, so we maintain the correct number of cells and we stay the same size, over 1 million cells kill themselves in our bodies every second of every day since the day we were born. And the process they use to do that is an inbuilt process that's evolved for the purpose of killing our own cells. Now, Robert Hooke invented the microscope in about 1660, the compound microscope, and he was the one who gave cells their names cells, because he was looking at sections of cork, the bark of an oak tree, and and it reminded them of the cubicles that monks would live in monasteries. Now, the cells in bark are dead cells, but they only fulfill their function after they've died. So trees, like humans, produce cells only for them to die as part of a normal physiological process. They died because they killed themselves. In 1842, Carl Vaux observed death of cells in the backbones or near the backbones in toads as they were developing from tadpoles into adult toads. So this was the first recognition that cell death could be a normal process in animals. So that's way back in 1842. But the most common term used, the technical term used for cell death or physiological cell death in mammals is apoptosis. And that term was coined by John Kerr. And this is a picture of John Kerr. John Kerr was a Queenslander, emeritus professor of pathology up at University of Queensland. He just died a couple of months ago. I think he was 92. But very nice guy, very insightful guy. And he published this paper in 1972 where they adopted the term apoptosis for this physiological cell death process. He'd actually been working on cell death for a number of years before, and he called it shrinkage necrosis. But with colleagues Andrew Wiley and Alastair Curry, they decided to call it apoptosis. But the thing is that this process of cell death had been discovered and forgotten and rediscovered and forgotten and rediscovered multiple times since 1842. And this is a graph looking at the number of publications on cell death per year. And you can see, it obviously doesn't go back to 1842, but here we have Kerr's first paper on cell death where he called it shrinkage necrosis. And there were very, very few papers mentioning programmed cell death or apoptosis. Here's where the term programmed cell death was adopted by Richard Lockshin, who was looking at physiological cell death in caterpillars. And here's 1972 when the term apoptosis was adopted by Kerr, Wiley, and Curry. Here is where programmed cell death was first described in worms by Bob Horvitz in his lab. And here's where the first mutant worms were discovered. This is when the SED3 and SED4 mutant worms were discovered, and I'll mention them more later. This is where the BCL2 gene was cloned by Yoshida Tsujimoto, and still very few papers on cell death. But since then, the number of cell deaths on apoptosis has grown more or less exponentially. And currently there's about 23,000 or 25,000 papers on cell death published every year. And that's because for three reasons. One is we now know the mechanism of, or some of the mechanisms for cell death. Secondly, we know that the mechanism has been conserved throughout the evolution of animals. Plants seem to have independently evolved mechanisms for cell death. And the third reason is we know that abnormalities in cell death can lead to cancer in humans. And so everybody became interested in cell death because if you can understand the mechanism, then maybe you can develop drugs to treat it. So going back, failure of cells to die because they have too much BCL2 seems to promote cancer and autoimmune disease, but BCL2 is the only known component, the only clone component of the cell death mechanism known in any organism. And how it was regulated and how it functioned was not known. So how does BCL2 work? So that's something to think about. Well, how do you find out how BCL2 stops cells from dying? The only proteins with structure similar to BCL2 were proteins with unknown function in viruses. But presumably the reason, so viruses are really just captured genes that are operating on their own. And presumably viruses capture BCL2-like proteins to keep cells alive because viruses need to take over a cell's metabolism in order to make new copies of the virus. And if a cell can detect the virus and kill itself, that can stop the virus from replicating. So there's a good reason why viruses might want to carry copies of BCL2-like genes. So how could other parts of the cell death mechanism be identified? And the clues came from this organism. And this is a roundworm called C. elegans or Caenorhabditis elegans. It's a worm that's less than 1 millimeter long, but it goes from a single fertilized egg to a wriggling worm. It hatches out of its shell and it goes off wriggling worm in about 14 hours. And then after about 2 weeks, it dies of old age. But people at Cambridge, so Sydney Brenner and John Sulston and Bob Horvitz studied the lineage of cells in C. elegans. So here's some notes from a paper written by John Sulston. So this is published in 1975. And so what he did is he looked down the microscope, and he had a video microscope. So he looked for those 14 hours of development of the worm in its eggshell. So the fertilized egg, he called, cell number 1, and then one, it would divide in 2, and one cell would go to one end and one would go to the other end. So he named every cell during the entire development of the worm. And you can see here some of his notes. So he says he's 6:15 and he's 6:25, and he says, it's just hidden away here. At 6:30, pumping about stopped, another cuticle loose, another cell death. And so here is his lineage of the cells in C. elegans. So he worked out the lineage of every single one of the 1,090 somatic cells that are produced during development in C. elegans. But he found that 131 of those cells, and always the same 131 cells, died at the same time during development. So, for example, if he studied this cell called P0.1, it would divide in 2 at this time, and then each of the daughter cells would divide in 2. One of the daughter cells would divide in 2, but the other daughter cell would undergo programmed cell death. And it was always the same 131 cells, always at the same time. So he called this programmed cell death. Now, one of the beauties of C. elegans is you culture them on a plate an agar plate where they crawl around and they eat bacteria. But if you put a DNA mutagen on the plate, you can mutate their DNA, and then you can find mutant worms in which things are abnormal. And some of these worms, they called cell death abnormal worms, or SED worms. And they got a number of lineages of worms that they called the SED mutants. So, for example, if you look at one of those 131 cells that's destined to die, it shrinks down and looks like a little button, and then it's engulfed by a neighboring cell, and then it's completely degraded. Now, Sulston and co-workers found there was a gene that encoded an endonuclease that cleaves the DNA, and if they made a mutation in that, then the cell was engulfed, but the dead cell corpse and the DNA of the dead cell still persisted within the engulfing cell. Many of these mutant lines, and again, it was many years later before they actually cloned and isolated the genes and found out what sort of proteins they encoded. Many of the genes were involved in recognition of the dead cell corpse rather than the death process itself. But the most interesting ones were called SED3 and SED4, and in those worms, there were no cell deaths. The worms ended up with an extra 131 cells, but interestingly, the lifespan of the worm wasn't altered at all, suggesting that programmed cell death and lifespan are independently evolved and have independent mechanisms. So that was known largely from work from Bob Horvitz's lab, and they'd isolated these SED3 and SED4 mutants back in 1986, and they could cross these worms with these worms and work out which is what's upstream and what's downstream. But it was years later before they cloned the genes and saw what was encoded by the genes. Now, at the time, so this is in the early 1990s, I was at Stanford in Irv Weissman's lab, and I ran into Stuart Kim. Stuart Kim had worked on C. elegans, and he learned techniques for making transgenic C. elegans. And I said to Stuart, look, I want to see how BCL2 works. I wish we could do genetics in mice like you can do genetics in the worm. And he said, well, he's just learned the technique to make transgenic worms. And so we hatched a plan to go for gold, to find out if we put human BCL2 into one of these worms, would it affect programmed cell death in the worm? And so we hatched this plan, and this is a fax. For those of you who remember faxes, and I'm writing to Peter Candido in Canada, UBC in Canada, because he had some nice vectors to express genes in C. elegans. In fact, there was one that was a heat shock vector where if you heated up the worms, that would turn the gene on. And so I said, greetings, I'm working with Stuart Kim at Stanford. We want to see if human BCL2 has any death-sparing effects in C. elegans. BCL2 is a human gene, may interfere with programmed cell death, blah, 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 implicated in lymphoma in humans. We need a construct or a vector that will allow expression in C. elegans. blah, blah, blah. The one that I think looks like it might be ideal is this one called PPD6-1. Can you send me some DNA? And so Peter Candido sent some DNA for that vector, and I put in BCL2, and then Stuart Kim injected that into worms, and we gave them a heat shock to see what would happen. And this shows a picture of two sibling worm eggs. So they're still within, the worm embryos are still within their eggshells. But these ones have been stained with a monoclonal antibody, because they're around now, against human BCL2. So you can see that this worm embryo has turned on the gene for human BCL2 and has a human BCL2 protein, whereas this worm doesn't. Now, the way that we did these experiments was we put the genes into a parent worm, and so it generated offspring because they're hermaphrodites, and so we didn't know whether the eggs were transgenic or wild type. So the experiments were carried out in a more or less blinded fashion. We also put in a marker so that when the worms did hatch, they would either crawl through the agar or they would spiral through the agar, so we could tell which were transgenic and which were not transgenic. So this shows some pictures of some embryos still within their eggshell. And you can see in this one, you can see 1, 2, 3, 4, 5, 6, 7 dead cell corpses. But in this embryo, at this plane of view, you can't see any dead cell corpses. So Stuart did the counts of the corpses and then put the eggs aside, allowed them to hatch, and then saw which ones were rolling and which ones were slithering. And this shows the results. Now, each little square or triangle or circle represents an individual C. elegans embryo. And these ones here are wild type embryos. And in fact, all of this was done on a SED1 background, so the cell corpses persist. But these ones are more or less wild type. And at this stage of development, you could see an average of 40 corpses within the embryo. If you gave these worms a heat shock, you get a wider distribution of numbers of corpses, but it doesn't really have much of an effect. If we expressed a control gene, this is the gene for beta-galactosidase, which makes the cells turn blue, and gave them a heat shock, again, it was the same as wild type. It's not affecting the number of corpses. Now, here are the offspring from a parent. Some of them are going to be wild type and some of them are going to express BCL2, but these ones were not given a heat shock, so they weren't expressing BCL2. And you see, again, an average of 35 to 40 dead cell corpses. But this is the money shot. The offspring of the same embryo, some are wild type and some express BCL2. So these are the ones who are spiraling and these are rolling and these are the ones that were slithering. So they're offspring of the same parent. They're all given a heat shock. The ones that expressed human BCL2, there was a greatly reduced number of cell corpses. So this meant that the human BCL2 protein, like a gear, was able to enmesh with the worm's cell death machinery, even though humans and worms, our last common ancestor, lived probably over 600 million years ago. So the mechanisms for cell death have been conserved still functionally for over 500 million years. And it means that the process of programmed cell death in the worm and the process of apoptosis occurring to 1 million of your cells every second, it has been conserved to large part over the last hundreds of millions of years. So these are some quotes from Bob Horvitz's Nobel Prize lecture that he gave in 2002. He says, the thrill of discovery in the path to scientific visibility. People sometimes ask, when does a scientist feel that aha thrill of discovery? In the case of our studies of programmed cell death, my biggest thrill was probably on February the 12th, 1992. That was the day Michael Hingartner obtained the sequence for another one of these SED mutants, SED9, and found that the most similar gene at the top of the list was human BCL2. We immediately realized that the pathway of cell death genes that we were studying in C. elegans was very likely to be similar to the pathway that controls apoptosis programmed cell death in humans. Now, you'll recall that we were doing these experiments at more or less the same time. We started them in July 1991, and Horvitz got the sequence for SED9 in February 1992. Now, soon after that, Bob Horvitz rang up Stuart Kim and said, look, I'm my lab are the ones who do cell death research in C. elegans, because Stuart was telling him, what we were finding. And he said, you should stop, encroaching on my territory, more or less. And Stuart said, oh, look, Bob Horvitz is the 3,000-pound gorilla in the cell death field, and so he doesn't want to upset Horvitz. So then we hatched a plan. What we would do is we would talk to my boss, Irv Weissman, who's also a 3,000-pound gorilla, but in immunology and stem cells, and get him to negotiate with Horvitz. So here are my notes from July 11th, 1992, where I've got make Irv, so my boss, ring Horvitz. And I wrote these notes. Because Irv said, he happily talked to Horvitz. He said, our experiments are really cool, because he didn't know about them until then. And but he said, give me some background, on C. elegans. So Stuart gave him a crash course on C. elegans, and I told him that make a note to Horvitz that we started our experiments in July 1991 before there were even reports of SED9 mutant worms. So there was no evidence even of a cell death inhibitor in C. elegans. And we were looking for death-sparing effects of human BCL2 in C. elegans, similar to the effect of SED3 and SED4. We were not looking to complement mutant SED9 alleles. We were looking to get downstream substrates of BCL2. It's impossible to look for sequence homologs, as sequences are not made available by the Horvitz lab. We'd be happy to collaborate, but we knew that Horvitz was collaborating with another cell death researcher in the United States called Stan Korsmayer. So Irv rang up Horvitz and spoke to him and said, look, we'll do just this one experiment, no more experiments involving C. elegans, but it would be unethical to stop these experiments because, that might delay identification of other human cancer genes. So, and so Horvitz said, okay, you can do this experiment, but that's it. So we finished the experiment. We sent them off to Nature, and in those days you had to mail things to Nature, and then it would take weeks before the honeymoon period. And so we got this note to say thank you for submitting the manuscript. And then a couple of weeks later we got this letter back, arrived on 12th of October, saying thank you for submitting your manuscript. Human BCL2 gene prevents programmed cell death in C. elegans, which we are regretfully returning with this letter without review. It's Nature's policy to return a substantial proportion of manuscripts without sending to referees, et cetera, et cetera, et cetera. And so that was the end of that. So then we thought, well, look, so the next thing that happened was in the Worm Breeders Gazette, dated October the 1st, 1992, Horvitz published that SED9 and BCL2 have sequence similarity. So we could clearly, that bolstered the evidence that our conclusions were correct. So we said, look, we'd like to bring that up with the editors at Nature so they'll reconsider our paper. But you see here that these abstracts in the Worm Breeders Gazette should not be cited in bibliographies. Material contained herein should be treated as personal communication and should be cited as such only with the consent of the author. So we rang up Bob Horvitz and said, look, can we mention as personal communication the fact that SED9 and BCL2 have similar sequence similarity? And he said no. And Nature said, no, they're not going to consider things any further. But that, again, is why it's great to be in the lab of the big honcho, because Irv, my boss, was a mate of the editor of Science. So we reformatted and sent the paper to Science. And here's the paper published in Science in December 1992, Prevention of Programmed Cell Death in C. elegans by human BCL2. The mechanism of programmed cell death controlled by BCL2 in humans is the same as that in nematodes. And that created quite a splash because it was picked up by the New York Times. Geneticists target immortal cells buried in the heart of body cells as a powerful gene that can make cells live forever, et cetera, et cetera. This gene is known as BCL2. So we're all on a real high after that. And then, so that was December 1992. And then in February 1994, Mike Hengartner and Bob Horvitz published the sequence showing that C. elegans SED9 encodes a functional homologue of the mammalian proto-oncogene BCL2. So this is published in Cell, and to give them credit, they say these results suggest that SED9 and BCL2 are homologues, and the molecular mechanism of programmed cell death has been conserved from nematodes to mammals. And they said These results confirm and extend similar previous observations of Vaux 1992, as we have directly observed extra surviving cells rather than inferring their presence from the absence of cell corpses. So there you go. And then here is Bob Horvitz at his Nobel Prize ceremony, because he shared the Nobel Prize with Sidney Brenner and John Sulston. And it was not just for cell death, but it was also for the process of development in the worm C. elegans. But the Howard Hughes Medical Institutes had a press release where they say Horvitz and his collaborators discovered that SED9, which protects the worm against cell death, is similar to BCL2, a human oncogene discovered by Howard Hughes investigator Stanley Korsmeyer in patients suffering from follicular lymphoma. Again, no mention of Sugimoto, who was the first to clone it, or Mike Cleary, who was the second to clone it, only Stan Korsmeyer, who was the third to clone it and cited all of the previous labs, and also cited our work in looking at BCL2 in FDCP1 cells. Unlike other oncogenes, BCL2 does not promote growth of cells. Instead, it blocks cell suicide. And when BCL2 is dysregulated, as in follicular lymphoma, it extends the survival of cells that would normally die. So in the worm, if you have a cell that's destined to die, it shrinks down like a little button. It's engulfed by a neighboring cell, and then it's completely degraded. Now, homologs of the worm genes were identified. So humans have a whole bunch of SED3-like genes, and they're called caspases. They're proteases that digest the cell from within. The worm has that SED4 protein, which is a homolog of mammalian APAF1 and an adapter protein. But while in the worm, SED9 directly inhibits SED4, and BCL2 is an equivalent of SED9, and there's a whole bunch of BCL2-like genes. In the worm, SED9 is antagonized by a so-called BH3-only protein called EGL1, and mammals have a whole bunch of these BH3-only proteins that inhibit BCL2 family members. In mammals, it's more complicated because BCL2 doesn't directly work on the adapters. BCL2 inhibits the action of pro-death BCL2-like molecules called BAX and BAC that allow release of cytochrome C from the mitochondria, and cytochrome C is needed to activate APAF1, although it's not needed to activate SED4 in the worm. So you can get clues from C. elegans, but C. elegans can also be slightly misleading because the wiring is different, has become different over those 600 million years. But, you know, the other lesson was that there are these important inhibitors of BCL2 called these BH3-only proteins. So failure of cells to die because they have too much BCL2 seems to promote cancer and autoimmune disease. So if you had a cancer characterized by too much BCL2 or an autoimmune disease, it might be caused by BCL2 stopping the cells from dying. What you'd like is you'd like to have a drug that could block BCL2. So something that worked like those BH3-only proteins. And that was what was developed at Abbott, especially by a guy called Steve Fezzik. But Abbott was collaborating with a company called IDUN, and I was a consultant for IDUN. And at one stage, I got this. So in 2001, I got this phone call from Tilman Altersdorf. And after getting this from IDA, and after I got this phone call, I sent this email to my ex-boss, Jerry Adams. And I say, this is what I recall from the phone conversation with Tilman Altersdorf from IDA. They've produced a number of non-peptide molecules that bind to the groove, it should be groove of BCL2 and BCLXL. So that act like BH3-only proteins. Of the two they are studying, one binds to both BCL2 and BCLXL, and one binds BCLXL only. The first, but not the second, will kill follicular lymphoma cells in vitro, but it hasn't been tested on other cell lines, but it's not toxic to fibroblasts, but they didn't have any in vivo tests. They were keen to study these compounds in vivo, so in mice, not in humans, to see whether the levels that are capable of killing BCL2 expressing cells are toxic to normal lymphocytes and other cell types. They're doing this work in association with Abbott and Steve Fezzik. Abbott does not want to use a validation system that would create problems with the patent on the oncomouse. Remember the oncomouse I mentioned before? That was a very broad patent that covered basically any mouse that contains oncogenes, but they didn't take out patent protection in Australia. So we'd be free to test in our transgenic mice these compounds. Tillman asked whether people at WEHI would be willing to try these compounds on BCL2 or MYC BCL2 transgenic mice, et cetera, et cetera. So I told him that we, in principle, would be very interested, and he said he'd call me again after they and Abbott had had a better look at the oncomouse patent situation. And that led to establishment of a bilateral agreement with Abbott, down below, he's Steve Fezzik, and Walter and Eliza Hall Institute to test their first compound. And the first compound was called ABT 737. And here it is binding into a groove on the surface of it turns out, BCL-XL. Now, it turned out, and this was, some of the beauty of the work done at the Walter and Eliza Hall Institute, they were interested in what regulates platelet numbers. And they generated a lot of mutant mouse lines that had abnormal numbers of platelets. And it turned out some of the mutant mice had mutations in the BCL-XL gene. BCL-XL is a BTL2-like protein that turned out to regulate platelet survival. And as I mentioned before, this drug that Abbott had developed inhibits BCL-XL as well as BCL2. So that gave us a heads up because Andy Roberts at the Royal Melbourne Hospital had started testing in patients this drug, or a derivative of this drug, but at WEHI, scientists were able to warn him to say, watch the patient's platelets, because these drugs might reduce the number of platelets and then the patients will be at risk of bleeding. And it turned out that they couldn't give curative levels of the drug to patients because their platelets would drop down too low. So that led to establishment of a three-way partnership, this time between Genentech, and this slide comes from Genentech, and the Walter and Eliza Hall Institute, and also AbbVie, which was formerly part of Abbott, to develop a new BCL2 inhibitor that was specific for BCL2 that didn't also target BCL-XL, so it wouldn't alter platelets. And this drug that was actually discovered in the United States at AbbVie was called venetoclax. And this is a picture of venetoclax binding into that same groove of BCL2, like a BH3 only protein. And this is from the New England Medical Journal, Journal of Medicine paper. Here's Andy Roberts, who did this, carried out this trial. So it was first used in humans at the Royal Melbourne Hospital, just behind us. And these are white blood cell counts. And so these are patients with chronic lymphocytic leukemia. And in chronic lymphocytic leukemia, you have very high levels of BCL2. And these black dots represent the initial white blood cell counts. You can see this break here. Here's the normal levels where the dotted line is. That's the normal level of lymphocytes, but these are the levels that some of these patients had. And the bottom, when you go down from that black dot down to that white dot, that was the lowest level they had after treatment with this drug, venetoclax. So almost every patient responded very rapidly to this drug. And some of the patients had ongoing responses. And here's a couple of patients, and they'd previously been through standard chemotherapy, multiple rounds of chemotherapy, and now they're resistant to the chemotherapy. And they were enlisted in this trial with venetoclax. And I believe these guys are still alive today. They'd been given just a few weeks to live, and yet their white blood cell counts went from greatly elevated down to more or less normal. Now, some patients have since the lymphocytes have come back and they've got a mutation in BCL2, so they're resistant to the drug, but there's ongoing development, and now companies have developed new drugs that also target the mutant forms of BCL2. So there's like an arms race, but every reason to be optimistic. And here's from Wikipedia. Venetoclax, under these brand names, is used to treat people with chronic lymphocytic leukemia, small lymphocytic lymphoma, and acute myeloid leukemia. And it's now more or less frontline treatment for them. It's on the PBS. It's approved by the FDA. And this research came from from initial studies, looking at cells, looking at worms, from tissue culture, and it's now led to this new treatment for leukemia. And indeed, evading apoptosis is seen as one of the hallmarks of cancer. And so I'll close now and thank lots of people, my supervisor, Jerry Adams, Mike Cleary for giving us the BCL2 cDNA, Wendy Cook, who did those experiments in FDCP1 cells, showing us a good cell line to look for cancer development, Ishwar Hariharan for the Zen vectors, Alan Harris for managing a lot of the mice. I should have written down, I'll have to modify this, Sue Barth, because Sue Barth made those transgenic mice at WEHI, got that whole process going. Andreas Strasser for his immunological insights to realize that cell death is important to avoid autoimmune disease as well as cancer. Stuart Kim for the worm experiments. Irv Weissman for all of his contacts and his influence with big honchos. And lastly, WEHI, AbbVie, and Genentech for translating these things into drugs that actually can be used to treat patients. So I'll thank you for your attention, and I'll try to address any questions you might have.